So, I told you about the nutritional value last time, this I am not going to discuss. We stopped here the biologically important peptides. See, there are many peptides which are there. I will ask you to note down only those points, rest are available in the book. So, these biologically important peptides, they perform important function that may come as a short note. One thing you note down, glutathione. Glutathione is commonly asked, it is a common short note, very important tripeptide. I do not know whether you are able to see from a distance because the picture reading quality is also bad. So, erythrocyte membrane integrity, its function, it is a tripeptide made up of glutamic acid, glycine and cysteine. It is a very important tripeptide. Many times teacher will tell you or will discuss in the class, at least these 4 points or whatever I have written in highlighted, erythrocyte membrane integrity to one function, keep the enzymes in reduced state, it is having a strong reducing property, protects the cell from stress by reactive oxygen free radicals, peroxides, and detoxification of xenobiotics means chemicals or drugs whatever we take. TRH, thyroid releasing hormone, it is a tripeptide. It may not be there in your book. TRH is produced by the hypothalamus and it is stored in the posterior pituitary. So, the function of TRH is to stimulate TSH to produce thyroid pituitary to release more TSH. Pentapeptide, Enkephalin, you write down. Enkephalin is very much important because it is an endogenous opioid. Endogenous means which is produced within the body. Exogenous means when we take from outside. So, in our body we are having producing neuropeptide, that is one neuropeptide, that is pentapeptide, that is called enkephalin. It is present in 2 forms. One is met enkephalin, one is leave enkephalin. That met means that terminal part, that 5th amino acid is basically met, and the leave enkephalin is 5th amino acid is leave. That is the difference. It modulates pain perception. Octapeptide angiotensin 2, it causes hypertension, and to prevent its formation drugs are used. So, that is important. Nonapeptide, oxytocin and vasopressin, these are the 2 hormones. Oxytocin, nonapeptide is oxytocin and vasopressin. Oxytocin function is, this will be told in your physiology class, it causes uterine contraction, and vasopressin prevents excess formation of urine. That means water is reabsorbed by the kidney, that is anti-diuretic hormone. It prevents diuresis. Diuresis means increased formation of urine. Bradykardin is another nonapeptide, it is a protein vasodilator. And decapeptide angiotensin 1, it is basically a precursor of angiotensin 2. So, from angiotensin 1, angiotensin 2 is formed, that means 2 amino acids are removed, then it becomes an active form. Okay. So, bioactive peptides are produced from the foods which we take, either as a protein form like milk, like fermented milk, from dairy products, meat, egg, poultry, many things we take, these are called bioactive peptides. The lists are many. Main functions, it can be obtained from animal, it can be obtained from plants also, and they perform lifestyle disease prevention. Lifestyle disease prevention, what are the lifestyle diseases? One is obesity, so it will act as an anti-obesity. Hypertension, So it will act as an anti-hypertensive agent. So coronary artery disease during there is a thromboembolic manifestation. So it prevents thromboembolism. Also it acts as an anti-inflammatory. So immunomodulatory. So these are the functions of bioactive peptides which we are taking, and that's why we are not suffering from all these things. If you do not take in adequate amount, obviously this thing will happen. Now, I mean, coming to today's topic is protein. Basically, I will be teaching you today structural organization of protein in detail, and that was left over. It can be told in 5 minutes, it can be told in 50 hours also. So, chemistry of proteins, one is amino acid. How is protein is formed? Protein is formed by the successive amino acids joined together by peptide bond. So, successive amino acid L-alpha amino acid. The peptide bond is formed between the L-alpha amino group and L-alpha carboxyl group by the successive amino acids. Okay? And then once it is formed, then it has to be in 3-dimensional structure. Then only it will be functional. And that is a tertiary form of structure. Okay? So, if I keep 2 amino acids together, will it form a peptide bond? If 2 amino acids are kept together in the test tube, will it form a dipeptide? Means will the peptide bond will be formed? What is your answer? So, peptide, there is a carboxyl group, there is amino group. So will they react spontaneously? Some enzyme is required. What enzyme? Enzyme will be required. You can synthetically prepare definitely peptides, but that is a lengthy procedure. In our body, how the sequence of the amino acids will be decided. The proteins will be basically a linear chain of amino acids. Now, how the sequence of amino acid will be decided? Genetic code, yes, by through mRNA, as per sequence of the mRNA. Okay. So, as per sequence, so it is genetically determined. So, during protein synthesis, during translations, as per codons, the amino acid will be placed to each other, and the successive amino acids will be joined by peptide bond using an enzyme called peptidyl transferase. Okay, peptidyl transferase is the enzyme which produces peptide bonds, like glucosyl transferase enzyme makes the glycosidic bond. So, the speed of peptide bond formation in eukaryotes is about 6 to 9 amino acids per second. In contrast, prokaryotes it is about 80 to 110 amino acids per second, because in humans the sequence has to be maintained very correctly. There must be adequate proofreading, because by chance if a false or improper amino acid is placed in the peptide chain, it will cause abnormal protein formation. So, that is the reason this step is very crucial, and the rate of this joining formation of peptide bond in eukaryotes is a little bit slower, much slower, and I would say, than prokaryotes. So, peptide bond, only thing, 3 or 4 things you remember. Peptide bond is a partial double bond, write down, partial double bond, number 1. Number 2, peptide bond doesn't rotate. Peptide bond by itself doesn't rotate, it is a rigid bond. And second, but the side chains of amino acid, they rotate. Peptide bonds doesn't rotate. And CN bond, that is peptide bond, is basically in trans state. These are the 4 things you remember. So, partial double bond, it is a rigid structure, doesn't rotate, but the side chains rotate, and it is the CN bond is in trans state. You need not require to remember all those things. Amino acids normally are written from this peptide chain, as I have told you earlier. So there are two basically amino acids, whatever I have written, suppose it is X, suppose it is Y, or it is valine, or it is maybe phenylalanine or tyrosine, you can say, whatever you see. What happens here, as per sequence of the mRNA codon, these are placed. So, by convention, what is present on the left side, it is the first amino acid of that polypeptide chain. The second amino acid during protein synthesis will be placed on the right side. So, this end is called amino terminal end or N-terminal end, and this amino acid is the first amino acid. As the chain will grow, so this will be the carboxyl terminal or C terminal. Okay? So numbering will be 1, 2, 3, 4, 5, 6, like this. Okay? And here the peptide bond will be formed by CO, and one H will come, one water will be out. So CONH bond will be formed basically. But remember, this bond formation will require energy. So it will not form simply by the enzyme. So it's an energy dependent reaction. So each bond requires ATP. So this part, again the sequence of amino acid is fixed. Suppose if I put it in the reverse direction, like 6-1, your date of birth, 4-1, suppose 2002. Now if you write 2002, 4 and 16, it will not carry no meaning. Same thing will happen here. So altogether a different amino acid will be produced. So that may not be functional at all. So any change, major change in major area will cause alteration of protein. So characteristics of peptide chain is basically, it's a normally linear chain, the way I have described. Now to describe the higher order of structure, we will write in this form so that you understand, because all the structures are not shown in the linear fashion. Okay? So, if the chain contains 16 residual sulfur containing amino acids, then it can form suppose in this structure there is one sulfur group, sulfur containing amino acid here, one sulfur containing amino acid here, and in the other chain sulfur is here, sulfur is here. So this sulfur it contains basically sulfur hydryl group, SH. Now what happens, these two bonds, this sulfidyl group can join together to form a SS bond, that is disulfide bond. Okay, that is intrachain, means within the chain. Intrachain disulfide bond. So in primary structure, two types of bonds are involved. One is peptide bond and another is occasionally, as per need of the protein, there might be a disulfide bond. Okay. Now, if two strands are there, it can form one disulfide bond here. It can form a disulfide bond between these two. This is SH group, this is SH group also. So again disulfide, this H will be removed, disulfide bond will be there. So from here it can form a disulfide bond here. So it depends as per the structure, as per the need. This will be an interchain, means two different chains are there. That's why it called an interchain binding sites. You see, all the parts of the protein are not functional. Like in antibody, this is a big structure of antibody. You can see that the antigen basically binds to this small region, this one. That is called antigen binding region. Rest part doesn't do. So that small part is there responsible, and you see there are globular structures like this. This is called domains. That structure I will tell you in detail. Only thing is that time is running very fast. So this part you need not remember because these are pure chemistry. So structural organization, write down these four parts: primary, secondary, tertiary and quaternary. Remember the spelling here, note the spelling. It's not quaternary, it is quaternary. So this is the structural organization and this is the sequence. Because without primary structure, secondary structure cannot be formed and tertiary structure of course is there. Most of the proteins are up to tertiary structure, and that tertiary structure forms the functional part of the protein. Only some proteins require quaternary structures, means association of two or more tertiary structure proteins. That is a quaternary structure. Now, this secondary, tertiary and quaternary structure, these are called higher order of structures. So, this is the level, order or level of organization. I repeat, primary structure is the linear sequence which contains peptide bond and disulfide bond. Once the primary structure is formed, then the protein starts folding on itself. It's a secondary structure, just like this. This is a polypeptide chain, whatever I say. Now it forms a structure like this. Now it forms a right-handed helix. How will you know it is a right-handed helix or not? That I am telling you. See, if you put your finger like this up and look at the finger, how it is moving. It is moving in this direction. So it is basically a right-handed helix. So if you put it here, it is on the reverse direction. That is a left-handed helix. Similarly, if I put this thing right down like I brought one screw, if you screw this thing downwards, this part you see hand is here, and but the other fingers are moving in this direction. So this is basically the right-handed helix. Important part is that what is denaturation? Denaturation, one term you note down. Denaturation is loss of higher order of structures. Like these three structures will be destroyed, but the primary structure will remain intact. Okay. Now, this loss of higher order of structure is called denaturation. Now, this can be brought about, which I have also done during my PG class. We have denatured by putting an extra, what are the bonds here? The bonds, write down 4 types of bonds are there which maintains these 3 structures. One is hydrogen bond, one is electrostatic bond. Sorry? You cannot hear. Primary structure, there are 2 bonds. One is peptide bond, one is disulfide bond. The rest 3 structures are maintained by 4 bonds. One is hydrogen bond, one is electrostatic bond, one is hydrophobic interaction, and 4th one is van der Waals force. All these forces will be true for DNA study also, DNA structure maintenance. Okay. These are non-covalent bonds. These are weak bonds. A covalent bonds, non-covalent bonds requires an energy of 1 to 5 kilocalorie per mole. In contrast, a covalent bond requires almost 80 to 120 kilocalorie per mole. So, this is very difficult to break, but this can be broken very easily. So, higher order of structures can be broken by alteration of little bit of salt or changing the pH, so that non-covalent bonds can be disrupted. But the primary structure is important, and it determines the higher order of structure. Now, if I remove that part, suppose a salt or pH, if you adjust it, so it can come back to its own position. So, that is called renaturation. But if the primary structure is destroyed, then it cannot be reformed. So, reformation is called renaturation. So, if you heat a protein like egg protein and make a coagulation, that coagulation cannot be brought back to egg's normal structure. So, that is an irreversible process. Coagulation is an irreversible denaturation. Denaturation is there, but it has destroyed the primary structure as well. So, that is the reason I have written it. So, importance of primary structure is that primary structure is a structure which determines the higher order of structures, means how it will fold. That sequence is very important. Normally, if there is any change in the amino acid in a non-important area, be it functional, be it structural, then there may not be any change. Suppose if there is a damage in the entire Santiniketan Medical College campus in the football ground, there may not be any change. But if something happens in your room or something happens while in this lecture hall, then there will be a problem. So, even a change in a single amino acid repeatedly, that sickle cell anemia example will come, and it is written in a book, that is why I am writing. So, changing of one type of amino acid with another, like here polar amino acid by non-polar, that will cause alteration of the structure. So, primary structure importance is there. It determines the unique or specific sequence of a number of enzymes, and it is determined by the corresponding gene. It locates the number of presence of disulfide bond because of a cysteine residue mainly, and also methionine, because these are the 2 sulfur containing amino acids, and it decides the higher level of organization. Left side amino terminal, right carboxy terminal, I have already said. So, represented just like this, that as I have explained here, that amino here and carboxyl group is here, or it can be written in this form or a single letter abbreviation, as I told you earlier, to make the space very short. So, the higher order of structures, note down this only part, that much will be, at least you must remember that the minimum information you must have. The primary structure is basically a linear sequence of L-alpha amino acids joined to each other by a peptide bond, and also if required by a disulfide bond also. Secondary structure will be basically localized folding of the primary structure in a geometric ordered fashion. Why I am telling you geometric ordered fashion? See, if I take this cotton, this thread you have seen, it is maintaining a geometric structure, specific geometric structure. So, this is a highly ordered structure. So, this is also a highly ordered structure. This has been wound in a very ordered manner. That is what is secondary structure. Then tertiary structure is complete folding. Suppose this has been definitely arranged in a group. Now, if I make it like this structure will be short. Many areas which were apart will be brought close together, and some part will be exposed, some part will be inside. Those parts which will be exposed will contain the polar groups, because in our body water is there. So, this protein molecule will remain in the solution. So, polar groups will interact and the non-polar groups will be inside. That will cause a hydrophobic interaction. Okay. So, that the polar groups will come during after this 3D assembly, which is basically complete folding and assembly, so that this structure, which is almost a two-dimensional structure, this is a two-dimensional structure. Now, this is also a two-dimensional structure, you can say. Now, once I make this thing like this is no longer a two-dimensional structure, and this is a three-dimensional structure, and these are the areas which will act as a domain. Means it will have, it will serve, it is made only for a specific purpose. So, it can be made like this, it can be made like this, depending on what we require. Like for structural proteins, we require fibrous proteins, we require like this. So, 3D conformation is most important in the tertiary structure, and that is the functional unit. I repeat it, I am telling you, quaternary structure means two or more structures if assembled together, which will form an oligomeric protein, then that is called a quaternary structure. Now, some proteins are functional only in quaternary form, not in tertiary form. But without tertiary form, quaternary form cannot be formed. Now, these quaternary structures are usually held again by all these four forces, that is non-covalent interactions, so that they are not joined together. Rarely disulfide bond can be formed, but forget about that thing. So, these four units can be dissociated also easily, but this dissociation, how these four units gets associated? Because the hydrophobic amino acids, they remain on the surface between the two subunits, but the hydrophilic side on the outer side. Do you understand? Suppose here if I take this one, so these are the two subunits. The hydrophobic amino acids will be in this side. Here hydrophobic units will be in this side. Now, they will be basically maintain hydrophobic interaction, and the polar groups will be on the outer side. Okay. So, another, since this is a very large molecule, so electrostatic attraction will occur between the positive and negative charge, hydrogen bonding will occur between positive and electronegative, and the van der Waals force are very weak atomic forces working between atoms. So, structure functional relationship, see, normal structure means normal function. So, subnormal structure, abnormal structure or no structure. means subnormal function or abnormal function or no function. If the polypeptide chain is not synthesized, that means there is no protein, so that function will not occur. Now, structural organization, suppose collagen, I am telling you, every third amino acid is a glycine, and collagen undergoes hydroxylation, and defective hydroxylation is there, that means collagen will not function properly. So, as I told you in the first class, second class, that protein is formed at translation, then it matures during post-translational modification. It is one of the stage function, that is hydroxylation, so that this collagen becomes collagen matures to become a functional protein. So enzymes, there are many areas. One is catalytic area. If that catalytic area, the amino acids, if there is not the proper amino acids, so catalytic function will not occur. Similarly, there is an allosteric site also, which regulates the enzyme activity, whether it will bind to the substrate or whether it will not bind to the substrate, or the allosteric site controls the substrate binding. So if there is any defect, so again the catalytic function will finally affect because of the improper substrate binding by the enzymes. So transport protein, so binding of oxygen by the hemoglobin. If that particular area is defective, then oxygen carriage will be difficult, and that's what exactly happens in hemoglobinopathy. Abnormal hemoglobin production in a vital area which binds to the oxygen, then or any other substance which the transport protein actually binds, that will cause abnormality. So primary structure determines by the number and sequence of amino acids, peptide bonds, disulfide bond. Secondary structure, suppose in this is a linear structure, the third amino acid structure is here, sixth is here, ninth is here. Now the moment we make it a helical structure like this, that will come a little closer. This is in the secondary structure. Now in the tertiary structure, you don't know something which is further apart. Suppose amino acid is here and the other amino acid is there. Because of the structure, these two things might come closer. Okay. So this is the structure, is basically the overall arrangement of the final protein which will be functional. And quaternary structure, as I told you, it is because of association of two or more units. So proteins, what we are showing or what you will be seeing in the books, really doesn't exist in that form, just like atoms. This whatever structure I have written here, it is not present in the form, in the nature. Electron microscopic structure of albumin is like this. Hemoglobin, it is having 4 units. If you take a picture, these 4 units will look like this, and not the way you see in the book. This is purely a schematic diagram to make you understand. So that's the difference. So this structure of insulin will not be really in this form, but to understand. So primary structure of insulin, this will be taught several times, even your familiar medicine class. This structure, the amino acid is composed of this, you need not write, this is available in the book. So then it is made up of 2 chains, only you have to remember this 51 amino acids, A chain 21 and B chain contains 30 amino acids. And as I told you, since there are 2 sulfur containing amino acids, so between 6 and 11 there will be an intrachain disulfide bond. And there will be 7th here, 7th position here. So 7, 7, disulfide bond interchain, and here is 20 and 19, so that is a 19-20 disulfide bond. So that is the primary structure. See, normally this is a human insulin. So earlier human insulin were not produced in adequate amounts because in the 90s, from 1900, recombinant DNA technology was there. That's why human insulin was produced by that method. But way back in 1922, when actually the experiment was done by a Banting based, it was the bovine serum, bovine insulin which was done. So bovine serum insulin molecular weight, if you look at this thing molecular weight, this is only defined by 3 amino acids, but it was more immunogenic. Then search was made for alternative insulin, and porcine insulin is differing by 1 amino acid and it is less immunogenic. That's why even when we were students, either porcine insulin means from pork, bovine means from cow. So insulin was purified from those animal sources and used to be given to the human beings, but that used to cause allergic reaction, immunological reaction. That's why human insulin was developed, and this is being used for last 3 decades, more than 3 decades. So pro-insulin is not produced in this form. There is a very complex procedure. It was a 51 amino acid, but originally it is synthesized as pre-pro-insulin. It is having a signal peptide, and that is first removed, the 24 amino acids. Then pro-insulin is formed, which is about 86 amino acids. So from pro-insulin, there is a connecting peptide between the 2 chains, that is called C-Peptide, and that is of 35 amino acids. Finally, insulin molecule of 51 structure is formed. The importance of this, that when insulin is synthesized, when one molecule of insulin is synthesized, one molecule of C-peptide will also be synthesized. So in the blood, if we measure insulin will be present in equimolar concentration with that of C-peptide. Equimolar means that Avogadro number, 6.023 into 10 to the power 23. Now, if you are diabetic patient, so why I am telling you, in future you will be required to do medical examinations. Like somebody is really diabetic patient. Now he has taken insulin and he wants to show that his blood sugar is normal. So therefore you have to certify that he is a medically fit person. And somebody who is really normal but not on insulin, you cannot differentiate. Now how will you differentiate between the two? And that case came while I was in my professional life. I was the chairman of the board, and the case that was a medically unfit case. Now she tried to prove that her blood sugar was normal. She is no longer a diabetic. I don't know where she has taken insulin. She can take insulin in the thigh, but I cannot examine the whole body. What I did, I agreed her statement with her statement, drew the blood and sent it for insulin and C-peptide examination. Now C-peptide was conspicuously absent. Now, if I take somebody who is not taking insulin, and if I draw the blood and measure insulin and C-peptide concentration will be there. Now if those who are not taking, a C-peptide will not be there, and that will conclusively prove that he is on exogenous insulin, not on endogenous insulin. Okay. So sickle cell anemia, as I told you, the valine replaced by glutamic acid replaced by valine. I am not telling you anymore. The secondary structure, as I told you, the staircase example, best example what I could see, that you see right hand and you move in this direction. That direction of the hand, that is the right handed helix. Now if it is down, again the direction of the finger. That you remember here, staircase. Best example, as you move upwards, you see the rotation. So secondary structure is basically, again I told you these 4 bonds. Remember these 4 bonds, which I told you it will be required in secondary, tertiary, quaternary structures. So it forms alpha helix. There are different types of secondary structure. The most common is alpha helix. The beta-pleated structure then turns, reverse turns, loops, bends, and helix. It will not be possible because time is running very short. So alpha helix I will tell in detail. So it's basically a spiral structure, just like a spiral staircase. So it's a right-handed helix, and only thing you will have to remember that it is 3.6 amino acids per turn, and there is a pitch length, and there is a increase in distance by 0.54 nanometer. It is a right-handed helix. These are the points, and it is stabilized mainly by hydrogen bonds. Hydrogen bonds is formed between the CO and NH group. This is main stabilizing factor is hydrogen bonding. However, other forces also act like electrostatic, van der Waals, and hydrophobic interaction. So proline is very important. Proline I have brought one structure. This is formed just like this one. This is a proline structure. Okay. So you see these are again joined together. It will form a structure like this. This is the structure of a triple helical structure of proline. I think you will not forget with this at least example. Okay, three different colors are here. Note down this part, hydroxylation of proline and hydroxylation of the lysine are the most important post-translational modification, and it confers rigidity of collagen molecule which is present in abundance. The most abundant protein of in the human body is collagen, and stabilization is done by the prolyl hydroxylase and lysyl hydroxylase. Remember, see here vitamin C is required, iron is required, and proline is converted to hydroxyproline and lysine is converted to hydroxylysine by the enzyme proline hydroxylase and lysyl hydroxylase. So that is how this structure is stabilized. Now all of you know during your school life that vitamin C deficiency causes scurvy. That you know. But what is scurvy? What does it occur? Why does it occur? See, vitamin C is required here for collagen formation. It is acting as a cofactor. So if the vitamin C is deficient, adequate amount of hydroxylation of proline and lysine will not occur, and so the collagen layer will be formed, but it will not be a functional collagen. The end result will be the scurvy. Scurvy is not seen nowadays even in our country, but while going through the journals, I came across a scurvy deficiency reported from London. So scurvy is deficiency of decreased collagen, functional collagen, because deficiency collagen is there, but it is decreased and a defective. So gingival hyperplasia, if you see this type of picture, you might see, but have a suspicion if you have a hemorrhage also, because vascular structure, this wall are also, there are collagens are present, so that structure will not be also proper. So there might be hemorrhage. So we cannot imagine that a vitamin deficiency scurvy will occur in United Kingdom, London. So the case was reported in 2022 from London itself in British Medical Journal. And you see, this is the baby presented with scurvy, and after 4 weeks of treatment with vitamin C, you see the improvement. So normally should not occur. If you are having a vigilant eye, then it will raise suspicion. Suppose, as I told you, that gingival hyperplasia along with the whole body examination. Normally we don't do, that's why we miss the diagnosis. This will make you alert that it might be something else. It's not that a simple case of tooth infection or gum infection. So this case we got here at Santiniketan Medical College in last year. This is a case of epidermolysis bullosa, likely to be a case of defective collagen synthesis. Now see the condition of the baby, it's a 5-month-old baby, collagen formation is defective. And that is the reason so many blisters are there. That baby was lost to follow up, otherwise I could have taken up and wanted to have a genetic study on this baby to see whether really it is a collagen. This is a suspicious case of collagen type 7 deficiency. This I will tell you in detail during collagen class if I take. So cases you will see, only thing is that you have to have a vigilant eye and you have to follow it up. Secondary structure is beta-pleated, means the structure which was a little bit compact in alpha structure will be somewhat spreaded, extended just like a tin sheet, or this harbonium part where the structure will be like this, but there will be pleating structure. So beta-pleated structure can be stabilized again by hydrogen bonding, and it can run in parallel direction or anti-parallel direction, running very short of time. It can take a hairpin loop, like one will move in this direction, the other will move in this direction. That means it can be parallel, it can be anti-parallel. So it can take a reverse turn, it can take a loop. Beta turns are there. So 310 helix, one note down here. It's a variation of basically alpha helix. Normally in alpha helix 3.6 amino acid residues are done by per turn, but here it is a 3 amino acid residue. That is the difference. It is a little bit more compact structure. And why this 310? Because 10 atoms in the ring is formed by making the hydrogen bond. That is the reason its name is 310 helix. Super secondary structures are basically cylinders and barrels. You have read about chloride channel, you have read about ion channels, and without this type of structure, cylindrical structure, ion cannot pass through. So proteins will have different types of structure depending on the requirement. So motifs, write down what is motif. Write down what is it, recurring pattern of sequence of amino acids structures in 3D form. In 3D form. That is what is motif, and that is important for biological function. So there may be a zinc finger motif, like zinc is there and it is having a finger like projection. I will show it to you. Suppose this is a leucine zipper motif. So it's a leucine is present at the 7th position, every 7th position, that's what it is called leucine zipper. And you see there are two ends. So you have seen in the conjugated protein, protein reacts with nucleic acid, so it will form a nucleoprotein. So in the nucleic acid there is a major group and minor group in the DNA. So these scissor-like ends at this end will interact with the major group and minor group. So proteins perform a variety of functions, like some proteins will combine with globin will be interacting with the ferroporphyrin ring of the hemoglobin. So its structure will not be the same. So leucine zipper DNA binding motifs are there. So motif, there are many types of motifs are there that you will see in the book. Only thing is that it is done because of the very specific purpose. Okay. So it is done basically for a specific purpose. So another is helix turn helix motif. You see here, it is a connecting peptide. It binds again with the DNA. But helix loop, helix motif basically combines with the transcription factor. When DNA, from DNA to RNA is formed, transcription factors are required. So that transcription factor binds with the DNA. This helix loop, helix motif binds with the transcription factor and connects it to the DNA molecule. So there are very many purposes. So structure is basically motif, and the motifs are different in different. It might be the same also. So it is basic structure of tertiary protein is basically in a protein, it can be an alpha helical structure, it can be a beta-pleated structure. Now all these things will be joined together and to form this type of structure or this type of structure. This can be a fibrous protein and this can be a globular. চলে এসছে স্যার? স্যার চলে এসছে? আচ্ছা, this type of a globular structure. Okay? এই জন্য করে গোটা গুটি পড়ে দাও তো তাড়াতাড়ি, আমি আর দুলে বললে। Okay? Now, quaternary structure, as I told you earlier, that several units, 2 or more units of tertiary structure, if it is combined together, then it is called tertiary structure. It can be a same structure, 2 strands, like two threads. It will be a homodimer, same structure. If it is of like this, hemoglobin chain I am telling you, 2 alpha, 2 beta. So this is a tetrameric structure, but this is a heterotetramer. It is not the same thing. So it can be like this, tetrameric, heterotetrameric structure. 3 different types of things. So it can be like this also. 2 similar chain, third one is different, fourth one is different. So it can be a homo, when the same structure is there, that is called homodimer. When 2 different structures are there called, it is a heterodimer. Hetero and homo, this means you difference whether it is different or not. Now tetramer or trimer, that depends on what type of units are present, monomeric units are present. And last slide maybe this part I will tell you during enzyme class. The CPK is having 2 units, subunits, LD is having 2 subunits, M and H, hemoglobin 2 subunits, and immunoglobulin are having 5 subunits or 2 subunits also, 4 subunits. The protein folding occurs. Protein require also some protein to fold, like one human being also require occasionally help of some human being, and these proteins are called chaperones. They assist in correct folding of the protein. So if there is a defect in chaperones also, so that will not allow proper folding of the protein, and that will again cause a disease. Okay, I wanted to show you the video, but now time is very short, I cannot do this thing.